The camera fades in on a future Earth. Silent. Post-human. Covered in ash and ice. Yet somewhere beneath it all, in the quiet cells of something not quite extinct, a code flickers to life. A chain of molecules, still holding a memory. Ours. Human DNA, just 4 letters. A, T, G, C. Yet somehow, it has built empires, painted cave walls, flown to the moon, and asked questions no other creature has dared. But how? What, exactly, is so strange, so miraculous, about the human genome, that sets us apart from the billions of life forms that surround us? Because our DNA is, on paper, nothing extraordinary. Chimps share 98.8% of it. Mice, more than 85%. Even bananas overlap by 60%. And yet, we write symphonies. Split atoms. Imagine gods. This is not a story about what DNA is, but about what makes ours different. And perhaps, what makes it dangerous. Because nestled in that double helix, may not just be the secret of our intelligence, but of our self-destruction. So let's start at the beginning. Before CRISPR. Before genetics. Before humans even knew they carried a blueprint at all. In 1869, a Swiss chemist named Friedrich Miescher isolated something strange from pus-soaked surgical bandages. A sticky, phosphorus-rich substance inside the nucleus of white blood cells. He called it nuclein. He had no idea. He had touched the code of life. But the road to understanding DNA was long and winding. Gregor Mendel, an obscure monk, bred pea plants in a monastery garden. He noticed patterns in their traits. Green or yellow, tall or short. He didn't know it. But he was discovering inheritance. By the mid-20th century, science had reached a breaking point. In 1953, Watson and Crick unveiled the double helix. A twisting ladder made of sugar, phosphate, and nitrogenous bases. But still, what made humans special remained a ghost in the code. The genome was a script. But no one knew how to read the story within. And the question that lingered, was uniqueness hidden in the genes themselves? Or in how we use them? And more troubling still, did we really understand what our DNA was capable of? The year was 1990. A global effort began. Massive. Expensive. Ambitious. The Human Genome Project. Its goal? Sequence all 3 billion base pairs of human DNA. For 13 years, labs across the world raced toward a genetic moonshot. And in 2003, they announced success. The entire human genome, mapped. But what they found was shocking. Of the 3 billion letters in our DNA, only about 1.5% codes for actual proteins. The rest? Dismissed for years as junk DNA. Vast, silent regions. Repeating patterns. Mysterious instructions. And then came the deeper puzzle. We aren't genetically exceptional. We have fewer genes than corn. Fewer than some fish. Our genome is efficient, not bloated. So where does our complexity come from? Scientists began to realize. It's not the parts, but the symphony. Regulatory genes. Epigenetics. Timing. Our uniqueness lay not in what genes we have, but in how they're turned on and off. And that raises a question far stranger than expected. If our DNA isn't that different, why are we? Compare our genome to that of a chimpanzee. Nearly identical. But zoom in, and subtle changes appear. FOXP2. The so-called language gene. HAR1F. A stretch of DNA active during brain development. A handful of regulatory sequences that changed the size of the neocortex. These tweaks reshaped our skulls, rewired our brains, restructured our vocal tracts. Not many, but powerful. It's as if evolution didn't write a new symphony. It just changed the tempo, the rhythm, the conductor. But buried in that 1% is not just intelligence, but instability. Our cognitive leap may have come with a price. Anxiety, schizophrenia, autoimmune disorders, cancer. Evolution gave us imagination, and along with it, the shadow of madness. So what if our uniqueness isn't a triumph, but a trade-off? And if that's true, then what else does our DNA hide that we've yet to uncover? Today, we no longer study DNA like archaeologists dusting bones. We edit it. CRISPR, Cas9, discovered in bacteria, acts like molecular scissors, allowing us to cut and paste genetic code. We've already cured inherited blindness, altered embryos, modified crops. But humans? That's a line we're inching toward. In 2018, a Chinese scientist edited 2 twin girls to be immune to HIV. The backlash was swift, global. Not because it failed, but because it worked. We're beginning to tinker with what makes us who we are. Intelligence, memory, aggression, longevity. If we can change those, are we still us? The irony is brutal. The very tool that allows us to understand our DNA may soon allow us to abandon it. But who decides which genes are worth keeping? And how do we avoid turning evolution into design? Is DNA enough to define a species? Or even a person? Identical twins share 100% of their genes, yet live vastly different lives. Trauma, environment, nutrition, love, all reshape gene expression through epigenetics. Genes are a script, but epigenetics is the director. A child born during famine develops a metabolism shaped by scarcity. A grandparent's trauma can echo in the grandchild's stress response. The genome is not fixed. It reacts. It listens. It remembers. And if our DNA is so flexible, so malleable, then what makes us, us? Is it the code itself? Or the experience written over it? And if we change the environment fast enough, do we evolve without waiting for the genes to catch up? In some places, parents are already selecting embryos based on desirable traits. Elsewhere, entire populations lack access to basic medicine. We're not evolving equally. We're not even editing equally. As genome sequencing becomes cheaper, and gene therapy becomes profitable, a new species may not come from mutation, but from privilege. Imagine 2 paths. One edited, optimized, and enhanced. The other left behind. Natural. Flawed. Human. Will we become casts of biology? Will future humans interbreed? Or see each other as alien? Speciation, once driven by geography, could soon be driven by wealth. So what does evolution look like? In a world divided not by oceans, but by algorithms? Biologists now speak openly of post-genomic humans. Entities defined not by DNA alone, but by artificial intelligence, nanotech, biosynthetic implants. Our DNA will still be there, but maybe as scaffolding, not essence. Already, synthetic biology allows us to print life from scratch. If DNA is software, what happens when we start writing our own code from a blank page? Could a future human be born without a single strand of natural DNA? And if so, would that being still be our child? Or the start of something new? Strip away the mysticism, and DNA is a molecule. But inside it lies the full inheritance of the cosmos. Carbon from stars, hydrogen from the Big Bang, patterns carved by time. It built the eye, the lung, the poem. Yet for all its power, DNA is delicate. A single mutation can cripple. A misplaced base pair can end a life before it begins. We are, all of us, improbable survivors, products of balance, chance, and a billion years of trial. So as we stand on the edge of rewriting that story, one question should haunt us. Do we understand what we're changing? The camera zooms out from a single dividing cell. A heartbeat. A breath. A whisper. From cave paintings to CRISPR. From bone tools to biotech. The journey has never really been about survival. It has been about becoming. Human DNA is not perfect, not sacred, but it is ours. And as we move into a future of engineered traits, synthetic biology, and designed intelligence, we carry one last fragile echo of the past. A code shaped by lightning, hunger, fire, and love. It is not the uniqueness of human DNA that defines us. It is what we choose to do with it.